This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1279, recorded on December 12th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And out here in Western Mass, it's cloudy and breezy and chilly. It's 34 Fahrenheit, 1C, but just feels kind of raw.
Here in Chelsea, it's 3 degrees Celsius and looks like pretty sunny. Still cold, though. Also joining us from College Station, Texas, Jolene Ramsey.
Howdy, and hello there. It's overcast here, 68 degrees Fahrenheit and 20 degrees Celsius, but I still feel cold.
But why is that?
Because she's a Texan and she can't handle temperatures under 70.
Is there too much air conditioning in your office?
I'm sensitive to the cold.
Yeah, well, I am too. That's why I always wear thermal turtlenecks now in the winter. People say, "Oh, what do you think? You're Steve Jobs?" I say, no, I'm cold. And these are Uniqlo. They're
those are good.
They're fleece on the inside and nice and thick, and they're not very expensive. So I have like 5 of them. I can wear one every day and then wash them on the weekend. You know, it's all
Strategic.
sorted out.
What is Uniqlo?
Uniqlo is an interesting South Korean company. Is it South Korean or Japanese? It's Japanese. Anyway, it's like one of the biggest retailers in the world.
Wow.
I think it was originally called Unique, and someone misspelled it, and the owner liked Uniqlo. So, And they have stores and they have mail order. Jolene, I got to tell you, this is so cool. You go and you pick a bunch of stuff off the shelves, and then when you check out, they have these bins. You throw all your clothes in, and it tells you the total.
Really?
There's nobody there to mark each one, scan each one. Somehow in this bin, there's a scanner that scans everything. Here's your bill. Just put your credit card in. They put it in a bag for you.
Is it based on weight or something?
No, I think it's based on the tags.
They scan the tags. Huh.
Wow.
That's
fun. It does
fun.
it automatically. It's really cool. They have some okay stuff, But it was a good article about the company in the New Yorker a couple of weeks ago, and it's very fashionable apparently, but it's not very expensive. Okay, also joining us from Austin, Texas, Rich Condit. I'm sorry.
It's about time.
Sorry.
I bet you those clothes, the tags have all got little RF chips on them or something.
Yeah, that's
Probably.
I was thinking.
But do you
NFC.
take them home?
You go to the library and you stack your books up on the checkout thing, and it knows what you got.
Well, I think it's some grocery
No, my
stores,
library out here in the sticks, we don't have that. It's still barcodes.
But the ones in the library get reused over and over. The ones in the clothes would just be one time.
Yeah, you would think it would be too expensive to do.
No, NFC tags are dirt cheap. They're I have a pack of like 200 of them. They're the size of a nickel, and they stick on as stickers. And it was like a couple of bucks, and that's retail.
Wow.
This week in retail.
Aren't there grocery stores where you can just push your cart through a scanner and it tells you the total?
Yeah. You see these bits in airport where you go through a kiosk and you just pick out stuff and then walk out the other side, and it charges you. I don't know how that works. At any rate, you need to know here that it's a little warmer than in College Station. Jolene, you'll be interested in this. It's 71 degrees. And it's partly cloudy. And I'm really disappointed because I was, I've even got long sleeves on and stuff. I was all set for another cool day. Tomorrow's going to be a nice day, which is okay. And then it goes down to the 50s and 60s for about 3 days. And then it's going up to like mid to high 70s again in December. We're going to, at the end of next week, we're going to Oregon. The motivation for many of the people is to ski on Mount Bachelor. Mount Bachelor is setting a record for opening late. I don't think they're going to be open for the Christmas ski. I mean, they got no snow.
They don't make it either,
welcome to our new world here.
Yeah.
Did I do everything? I
You
think
did
so.
it all.
Yeah.
Yep. If you enjoy these programs where you hear science, which is very hard to find these days outside of journals. Well, you certainly can't listen to the CDC and the NIH and the FDA much. But we tell you science, real science. And if you like that, we'd love your support to do these programs. You can go to microbe.tv/contribute. Now, ASV 2026 is still months away, but we're going to start talking about it because there are deadlines. Current ASV members should have received an email about a week ago reminding you of deadlines for the meeting. And this meeting is going to be in Minneapolis at the University of Minnesota, July 27th through the 30th. I liked I like ASV in Minneapolis. It's a good place to have ASV. It's a really good city. It's a really good state that's been trashed lately by the president, and it's none of it's true. Okay, Minneapolis is a wonderful place. Of course, nothing of what he says is true. July 27th through 30th. So if you have to, you should renew your membership, right? It's asv.memberclicks.net. You can renew your membership, submit an abstract, apply for a travel award, and get meeting details. The abstract meeting is February 2nd, 2026.
The deadline, yeah. And that's coming up soon.
It is.
That's
Travel
why we're
award
going to
deadline.
flog this on you early.
Travel award deadline is February 9th. You must be a current member. Don't procrastinate, and the fees are not so bad. You know, it's very interesting. At this time of year, I always would forget to pay my annual membership, and then I finally said, screw it, I got a lifetime membership. I don't have to worry anymore. It's really good. So anyway, go to the asv.memberclicks.net. You know, we'll be doing a TWiV from ASV as usual, and Minneapolis is a very nice town. You get a lot of walking in. The campus is big. But I do remember one of the buildings is called Vincent Hall. Really
Cool.
well named, right?
Yes.
Well named hall.
So it's Minneapolis, but it's a big campus.
I got it.
Yeah.
Okay. Alan, did you put the news in?
I did not. I think Jolene put this
I
in.
did.
Please
Yeah.
tell us.
But I did see this in my regular feed. But go
Please
ahead,
tell
Jolene.
us about it, Jolene.
Yeah, so today there was an announcement from the WHO, their Global Advisory Committee on Vaccine Safety, which has been around since 1999 and regularly reviews the safety of the vaccines that are available, reviewed the evidence again. That would be 31 primary research studies between January of 2010 and August 2020 from many countries around the world that support strong positive safety profile of vaccines used during childhood and pregnancy and confirm the absence of a causal link with autism spectrum disorders. And they additionally provide us with this nice piece of information that during the past 50 years, while vaccination has been available, childhood immunization has saved at least 154 million lives, which is a lot of people.
Yeah.
So this little announcement came out today.
So, of course, Junior would say, we don't need no experts.
Hello.
Yeah.
Is anybody listening?
Yeah, I mean, he's going to say that these experts don't know what they're talking about. They're paid by pharma, blah, blah, blah. But this is all good stuff, and we've known this for years.
Yeah.
You know, it's funny. On the page of this is news, medical life sciences. There's an ad for rapamycin. Gosh, it says it's the most promising health span promoting intervention. If you want to live longer, you just take rapamycin. We used to use it in the lab as a drug to do things to cells.
Yep.
Oh my gosh. Target of rapamycin. Remember mTOR?
Yep. Yeah, that's become a hot topic in longevity research.
Yeah.
People going on rapamycin.
Oh dear. Anyway, that's
So
good
I'm
to
supposed
hear
to
that.
take rapamycin just for kicks?
Yeah.
Apparently it doesn't hurt.
right?
At low doses.
Low doses.
I'm sorry. I'm all natural.
That's fair too.
I take no drugs and no supplements. I'm very fortunate.
If many people live into their 90s, and I think if you take this, I don't know how much longer you're going to live.
I'm not sure I want to live that much longer.
Well,
And
a lot
who
of
would-
the kind of the legitimate focus in longevity research is not so much on making people live to 300. It's on increasing what's referred to as the health span. So
Yes, right.
you still live into your 90s and then croak, but you croak in good condition. You are able to, enjoy your time more toward the end because you're not sick with a whole bunch of different things. But I will point out some of the things that have come up. Rapamycin gets hyped because it's a drug and you can sell it. There are a couple of other drugs that have been hyped for this, but there are other interventions like regular exercise and eating a variety of foods that are minimally processed that are like way, way up on the chain of effects in health span promotion. So you can do this on the cheap.
So it's an inhibitor of mTOR, which I knew, which is a master regulator of cell growth, metabolism, and immune function. So if you get an organ transplant, they give it to you to immunosuppress you. You must give huge doses.
That's a much higher dose than what's recommended or being promoted for this other use.
And
I'm not doing it.
by inhibiting mTOR, rapamycin mimics the effects of calorie restriction, a known lifespan extending intervention.
So you could just stuff your face a little less and
Take a walk.
I don't know about calorie restriction. I'm
it
not
depends on
really
how you
into
do
that.
Take a
But
walk.
exercise I can do. I can do exercise. And I can eat good food.
Yeah.
Oh, well, this is the world we live in. Okay, let's do some papers. Jolene has a snippet.
These are nice digressions this morning.
Yes.
For this afternoon. We're
Why
really
are they
doing a
nice?
good job here.
Well, I mean, I think I'm curious. I see an ad for rapamycin. I want to know
Absolutely.
Yeah.
what's going on. So,
Sure.
I used to use it in certain experiments in the lab, and I'm very surprised to see it being used to live longer.
Yeah, and I happen to have covered some research recently that touched on this very topic and talked to people who are who are researching it, and
But that's
There you
what
go.
increasingly these podcasts, we always diverge when we see something we're interested in.
Oh, yeah.
Hey, how does that work? That's the way it should be.
Yeah.
It's
Well,
we're
that's-
real people interested in the real life applications of science and science adjacent things.
Yes.
and that's actually one of the paper we're going to be talking about today. There's a little bit of that going on. Somebody set out to study something different and
Yeah.
ended up in a different place from where they expected to be.
Okay, Jolene, go for it.
Okay, so today I picked a different kind of paper on the topic of phage, and this comes from Nature Communications, just published online in December of this year, 2025. It's called Considerations and Perspectives on Phage Therapy from the Transatlantic Task Force on Antimicrobial Resistance. There are representatives from the different countries that are involved in this transatlantic task force, from the US, from the UK, from Norway, from the Netherlands, from Belgium, from Canada, from different parts of the European Union as well. And these individuals co-authored this report based on a meeting that they had in November of 2023. So this transatlantic task force was put together to help promote communication and strategy sharing to combat global antimicrobial resistance, which has become a major issue in recent years. So as a kind of overview, Phage therapy is something that people are very interested in using. Currently, it's only available as an experimental therapy. And so the question is how to move past that model so that this can be regularly accessible to individuals in these countries and outside these countries. And this is really kind of an overview of the regulatory frameworks that are currently available in these places. And so we're not really going to be talking about the science of the study of phage, but more thinking about how our countries going about trying to use phage medicinally. The idea is by sharing experiences, we'll be able to combat antimicrobial resistance more effectively on a global scale rather than not working together.
And I just want to point out a couple of things that struck me here. First of all, it's open access. And secondly, if you're a policy wonk or interested in science policy, this is a really kind of chunky, interesting problem to get your mind around. There's a lot going on here. Back to you.
Yeah. So I wanted to give just a tiny bit of historical context for this report because this is where we are today, but where did we come from? So phage when they were first really discovered and talked about and used in this way back in 1917 by Felix Durrell, we didn't really know what they were. We just knew that some applications of them seemed to cure diseases like dysentery in soldiers. And there was great interest in applying them, but people didn't know what they were, again. So they applied them very sporadically and not as logically as we can today because they didn't have enough information. And in 1928, penicillin was discovered. So that started the small molecule antibiotic kind of development in parallel. And so Felix Durrell really was promulgating and trying to get things to work. He did cure people of things like the Black Plague and other stuff like that. So it was working, but it wasn't always working. And so there were a lot of people that were fighting back and forth. It was confusing. And in 1934, the American Medical Association commissioned a report that would kind of pull all of this information together in a comprehensible way to get an idea of whether or not these phages were going to be useful in treatments of people with bacterial diseases. And kind of just like a little side note on the Durrell side of things. So he was quite a character, and he was what is called a congenital contrarian. So he was hired by Yale in 1928, the year that penicillin was discovered. And then he was fired 5 years later. And the person who co-authored this report commissioned by the AMA is the person who replaced him at Yale. So Stanhope Bain-Jones. There was a series of articles. They pulled together lots of reports. Some were positive, some were not. They overall concluded that there was a mismatch between phages and bacteria, not enough animal models, too many failed experiments, lack of controls. So over thousands of patients in more than 70 studies, the therapeutic value was inconclusive or negative.
And I want to point out for longtime TWiV listeners, if the name Durrell rings a bell, it's because we talked about the original discovery of phage with Durrell and Twort many, many episodes ago.
Was that that was the Invincible TWiV? Is that right?
I think it might have been.
That was you and me.
Yeah.
It was.
And we did the Twort paper.
Yeah.
I was looking for that. I'm going to look it up. Invincible.
It's
way back. Yeah,
yellowed.
I'll look for it.
It's
Okay.
yellowed.
So their only positive outcome towards phage for this report was that it does work, or it did at that time work with local infections in staph, like boils and furuncles. They had 15 studies with over 2,000 patients that were basically all positive. And so fast forward about a decade, the West started using antibiotics. We got into a world war. Phage were seen under the microscope, and we started to understand more about what they were. In 1941, the AMA commissioned another report, and they kind of concluded that phage-induced lysis of bacteria played a negligible part in their clinical application. They said that they might be good because they had the lysate components induced an immune system. And so phage basically became part of the molecular biology revolution. And that was the majority of what they were studied for in our part of the world. And there have been since then still people who have tried to apply them, but it was never systematic or on a large scale. So now today, for the past about decade, people around the world in many different countries have put a lot more effort into developing phage as alternatives to antibiotics to treat antibiotic-resistant infections. And this is where this report comes in. It kind of, again, starts to pull together what's the state of affairs in a lot of places where there is great interest in applying phage therapy. And they give us several examples that they had in their meeting, including the idea of having what is called the magistral phage strategy that is being used in Belgium. And I'm not going to talk a lot about it, but in TWiV 1153, viruses that can make you well, we talked to Christina Dombrovska and Rob Levine about this model of giving people access to phage under the auspices of a pharmacist and medical provider. And they also talk about both individual emergency use cases of phage being applied to patients who have no other options, as well as potential for randomized controlled trials. And we did talk a little bit about this. We talked a lot about an individual case in TWiV 502, Texas road phage, where we talked to Jason Gill and Rai Young about the Tom Patterson case. And we haven't talked a lot about the ongoing phage trials that are currently in progress, but those are actually registered on clinicaltrials.gov. And depending on the search terms that you use, yesterday I was finding something like 128, or if you put in some filters, then it's more like maybe 70 or something like that are ongoing. But many of those trials are single patient trials and not kind of larger studies. So in addition to those human phage use concepts, they talk about this other development, which is the use of phages in veterinary applications. And so this is really interesting. People have been trying to use phage to treat bovine mastitis in dairy cows for a long time, and there's interest in using them to treat skin infections in cats and dogs. But a place where they're actually having a big impact is in fish farming. So in Norway, where they are farming salmon, they actually already vaccinate their fish against a bacterial disease and other things. And when they are vaccinated, because the fish are young, that stresses them and they're more susceptible to a particular infection with Yersinia ruckeri. And so what they do is they started putting phage into the tanks that the salmon swim through on their way to the vaccination process. And that has been helping them to lose less fish during their vaccination treatment.
I think that's a great story. I love that.
Yeah, and what I
really
thought
cool.
was interesting here is from a policy perspective, the reason they're able to do this is because the phage is considered a water treatment.
Exactly.
Not a fish treatment. Because the fish are just swimming through the water that has this phage in it. You're not dosing them with it, and therefore it gets around the veterinary medicines guidelines. You don't have to have special approval for it. And that's kind of a recurring theme in this whole discussion, is that there's this chicken and egg problem that you can't implement something on a wide enough scale to really test it because the regulations forbid you from doing that, but you can't get the regulations changed because you don't have the data from a large randomized controlled trial.
Yeah.
So this was a unique situation where they're actually able to deploy it.
It's really great that it's working for them. And they also said that they've tried to expand it into use for farming rainbow trout or for farming salmon in different geographical areas, and it hasn't worked. And that highlights this continual issue that we have, is that phage are very specific to a particular bacterial host. So unless it's the exact same bacterial host causing a disease in another geographical area, that particular phage won't work.
And I see at the mention of rainbow trout, Dixon is looking pretty bright in the background there.
Yeah,
Rich.
Oh, yeah.
perked right up.
Yeah.
Nice. So then what they do in this paper here is that they split up their explanation according to the 4 major areas that TATFAR, or the Transatlantic Task Force, covers. So I was just going to kind of briefly go over each of the areas because I think there are some themes and some interesting little tidbits for each place. The first one that they talk about is the European Union. And for each of these, they have a particular legal way that they have to define phage, and that determines how they're regulated. So the European Medicines Agency classifies phage for treatment as biological substances, and the medicines that contain those phage are considered biological medicine products. And so those have to be characterized. The phage themselves have to be characterized for them to go through their authorization procedure. And that has to be done for each developer of a particular product. At this time, there are no approved phage products in any of these areas that I'm going to be talking about. But they say that some of the regulatory considerations that are important to consider as they're picking out phages to try to use is that they have to be identified, pure, potent, and viable. And so they have to have a bank, basically, of these that are available and ready to use. And they also have to have the host banked, and that host has to be characterized so that it's not something that's going to be spreading antibiotic resistance genes or virulence factors. And they say also has to be screened for prophages, which I think is very tricky because all bacteria have some prophages in them, and we can't tell just by looking at them what their activity will be. But the even most tricky part is that most of the time, as is the case for this EU situation, is that they really want final drug products to comply with good manufacturing practices. And that is something that is a little bit expensive. So GMP, as it's called, is really a quality measure and a quality control so that there is consistency in the production and testing of any product that's going to be used in humans. And it requires validation, extensive documentation, and those are usually required for something to, you have to have that process in place for something to go through an approval process. And so in the EU, they require new products to be compared to what they call the BAT, or the best available therapy. And they often even require, when it's being tested, that they show superiority over the best available treatment. So they'd have to have combination therapy with the best available treatment and then show that on its own it can work better than the best available treatment. And so those things are kind of what's guiding a lot of what's going on in the EU. But there's one additional aspect that I think was particularly emphasized here, and that's their regulations on genetically modified organisms. So because in many of the individual cases that have been successful for phage therapy, they have changed what would typically be a temperate phage, or one that could integrate or maintain itself in the bacteria without immediately killing the host. They've changed those to be virulent or kill the host right away. That's considered engineering or genetic modification. And in the EU, they have very strict guidelines on use and ability to market those kinds of products. So they say that phages would have to comply with those regulations. And
see, they should listen to TWiV so that they know that the phages are not actually living organisms.
So wait a minute. Can you take cloned insulin?
In the EU?
Yeah.
I think that's not considered a genetically modified product.
It's not, right. So the product there is the protein.
Right.
The product is not, you don't get a needle with the bacteria in it or the yeast or whatever it's being produced in. So the production process uses a genetically modified organism, and that's considered okay. But the product itself, in the case of phage therapy, you would be releasing into the general environment. So if you've got a fermenter and you're making insulin, you purify that out and the organisms stay behind. If you've got a phage that you engineered and you're calling that your genetically modified organism, you're going to put this into a patient and it goes out into the world, and that's what the EU regulations have a problem with.
Mm-hmm.
Okay.
Apparently, they recently released a chapter with guidelines on phage therapy medicinal products that have flexibility in them with regards to phage. I have not read that, but that is apparently available to help guide the development of these products. So in the EU, individuals can access unlicensed phage products because they are. I mean, this is really where most of the success has occurred in the past 100 years, is the ability to use things like is going on in Belgium, in Poland, in Georgia. All of these countries have had processes for giving phage to patients. And the problem is that the frameworks that are being used don't really facilitate the collection of the large-scale trial data that would give greater access over their broad populations. So the way that I summarize that is that it helps us save lives now, but it doesn't help us in the long term with having access for more people. So that's the EU. And then the next country is Canada. And this is all through Health Canada. I think that here, their really big focus is on both preserving the effectiveness of current antimicrobials. So they're emphasizing both extending the lifetime use of antibiotics as well as facilitating access to phage. The major difference I see in Canada is that although they kind of have a way to give phage to people, it's really basically by applying for single patient clinical trial authorization. And that is something that I found that this was really focused on kind of encouraging individuals to know what is needed to apply for a larger clinical trial authorization. But I looked through all the ones that have been done so far. They're all single patient clinical trials, which doesn't allow them to collect enough data to really improve the long-term access. And I think that is because of the extensive amount of information that you have to have on what you're planning to do and the plan that is required for that application. So there's something called their clinical trial application. They do have apparently a no-cost free advice consultation available with the Canadian authorities that go through these applications. And they've seen the single patient trials have occurred on all different kinds of infections, but that still, it requires a lot of information. I made a note here that in urgent situations, they only require phage history, lytic activity, potency, impurities, sterility, endotoxin level, and critical manufacturing processes, control, storage, and transportation, which if it's an emergency situation is a little bit of information to collect. But it says in less urgent situations, they have to have much more extensive characterization of the phage themselves before they can be submitted as part of this clinical trial application process. And that's the only way that they can get access in Canada, because the criteria are that people have to have a life-threatening condition, have exhausted all other treatment options, and they don't have access to the treatment any other way.
And this is a recurring problem with making policy about this, because right now what's happening is this patient or that patient or, somebody in Toronto and somebody else in Winnipeg, and whenever it happens, you get an infection, you're treated with antibiotics, they don't work, you get second line, third line, and finally your doctor is desperate to try to cure this infection that's antimicrobial resistant, that's, not responding to anything. And a researcher has available a phage that they think might work, and so they throw together one of these emergency authorizations and get it to the patient, and sometimes that works. And that's great if you're that patient and you're now alive instead of dead. But that's not how we do science. You can't really say, well, then you can roll that out for the population because you don't know. And that's the access issue you're talking about. You don't know that this actually works or if you're just lucky.
I was going to say, you give this therapy to them and it works. I would say you give the therapy to them and they get better.
Yes.
You don't really know if the therapy
exactly.
works.
You don't know if the therapy worked. You know that they got better.
So I don't know if this is the right place to put this in, but one of the big messages I got from this was that there's a big difference between therapies that are essentially personalized, where all these different infections and circumstances are different. So you may use different phage depending on the circumstance, and it's hard to do a controlled study under those situations. Whereas there are other diseases where, and they cite cystic fibrosis, where the infection is pretty much the same all the time, and you can come up with one treatment and do a lot of people, and then it's easier to control. But these personalized things, it's really difficult. And that gets me into, how do you do this anyway? I mean, so spinal surgery, for example, you don't do a random controlled trial, right? Where you cut somebody open and don't put in any hardware or anything and see how they do. It's a different kind of study. So it's no different than what's been going on in medicine for a long time.
Well, part of the problem is if you have patients who are acutely ill, right? Right. So you can treat one or two, but to collect 1,000 of them is not easy, right?
Yeah, you would have to do at least a large nationwide trial where you would just wait around for people to come through with a particular, because it's phage, it's species specific. It's got to be that species of bacteria that you're targeting, saying, okay, you've got that and it's multi-drug resistance and you've, failed all the first, second, third line therapies. And now you're eligible for this trial.
Right.
And that's one patient. And then you're going to have to wait around for somebody else to pop up like that. And that's a huge thing to coordinate.
That's part of, that's one other thing that they have, I think, is a strength in Canada's section that they talk about the establishment of banks. It's not that other places aren't doing this. It's just Canada has a long-standing phage collection that's actually named after Durrell, and they allow the acquisition of phage from inside and outside Canada. And so this kind of communication between countries allows for sharing, which is really helpful when you have such a very specific kind of infection treatment protocol needed. And so they're trying to establish protocols so that can be quickly and easily shared between centers. So for the UK, I think the one kind of big difference that I see here is that they're trying to come at it from a One Health angle, they say. And so they're taking into consideration that it's not just about humans, but it's about the environment and the animals that are in the environment. And you have to take that all into consideration to get these things to work together. And in the UK, they came out with a set of modified regulatory considerations for therapeutic use of bacteriophages in June of 2025, which supposedly has a comprehensive regulatory set of informations that tell the people who need to know how to develop and use phages in patients, and that includes licensed and unlicensed routes for both clinical trials, which they call like specials in their lingo, as well as importing phage from other places. So they are classifying phage as a biological product, and they do also have some special considerations for phage that are genetically modified. But they say they have separate legislative requirements that help to govern those. And then I think the only other thing I wanted to highlight from the UK is that they have this exemption that allows pharmacists to compose phage or phage cocktails to give access to individuals that are under the care of a pharmacist, under, I guess, being followed by a pharmacist. And that's because they have a separate governing body that makes sure that pharmacists are kind of following the rules and doing what they're supposed to do. So this is kind of another example of a way that there's access, but it doesn't contribute to the long-term data that is necessary for the acquisition of randomized clinical trials. And the one other thing that I'll mention for the UK is that they talk about, and this is something that's come up on TWiV before, their National Institute for Health and Care Excellence, or NICE in the UK, they have started exploring how to make phages available as prescriptions through a subscription model so that it doesn't just cover traditional antibiotics, but can expand eligibility to help cover phage and microbiome treatment. And so they're at least looking towards how to make the cost something that is possible for other people, because personalized medicine is something that is not cheap at all. And then the last country is the US, which in the US, phage products are governed under the FDA, or the Food and Drug Administration. And everything that we have access to right now, whether it's through a randomized clinical trial or individual treatment, comes under an investigational new drug application. And those are something that the FDA has been developing a feedback process and a lot of paperwork. I think that the US paperwork that was described in this review or perspective was the most extensive. They call it Chemistry, Manufacturing, and Controls, or CMC. And this applies to individual phage as well as combinations of phage or cocktails. And they have a very long list of things that must be known or tested, although they say that there can be some exceptions depending on the urgency of the review that is requested. And so I think from there, I just want to move to the idea of the challenges that they're summarizing for advancing phage therapy. And they categorize them under therapeutic research and development. So there is a lack of standardized assays and models that can help to demonstrate efficacy. There's not sufficient financial investment for the type of infrastructure that is going to be needed to deploy these at a large scale, and that we need standardized libraries or banks that are available to be shared and that can be quickly accessed when needed. And the second challenge category is manufacturing process. So it's one thing to make enough to do some lab experiments to test if the bacteria from the patient is susceptible. It's an entire other thing to scale it up and purify it away from any components that could further stimulate a harmful immune response. So there's that. And then there's the cost of doing good manufacturing practices, whatever the current state of that is. The third challenge are clinical studies. They say that we really need rigorous clinical trials that are completed and shared so that the data that everyone would like to see can be collected and analyzed for efficacy. And that way we'll know which diseases phage therapy is really going to work for and which ones it's not going to, because it's not going to work for everything. They talk about need for awareness among healthcare providers. And being not a healthcare provider, being a scientist, I get very excited about these things. The students get very excited. Individuals who could potentially use it get very excited. But we really need the physicians, who are the ones that have to put together this information and apply for approval from the respective government agency to be able to give it to the patients, to be aware and willing to work with this kind of experimental treatment. And then fourth are regulatory pathways. So basically, everybody has a different regulatory framework, and that does make the legal aspects of intellectual property, potential for a market that can sustain a company or government office that is producing these, and access for individuals to be very tricky. There's a lack of criteria that account for the difference that phage are as a potential medicine compared to all of the other kinds of products that are on the market, because they change. That's just, once you let them go, then they're not necessarily just going to stay the way that they were when you put them in, unless they're genetically modified. And even then, things you don't expect might happen. The last thing that they name as a challenge is the sustainability of the pipeline, which includes trials, accounting for market demands, and having economic models that can reward the production of these kinds of products to combat antimicrobial resistance. So kind of in summary, they say that although there are more than a dozen ongoing clinical trials between phases 1 and 3, there are significant obstacles that require us to work together to make this something that can be accessible around the globe and can be cost effectively used to combat antimicrobial resistant infections.
So what do you think, Jolene? Does this work?
I think that if we can focus on some areas that have shown promise and put investment to push that forward in a way that draws on the experience of individuals who are doing this in many different situations, then we will have very specific scenarios where phage therapy is possible for many people. But if we spread ourselves out too much and our messaging is wrong, that like phage are going to fix the antibiotic problem, then we're going to be hampered.
So focus on a manageable example where you can actually demonstrate efficacy convincingly to get everybody on board, and
work out the regulatory side. I think that continuously has to be a conversation, because we can make the most beautiful product in the world, but if we can't actually market it to the people who need it, then it's not going to be available here.
And money is going to be a huge problem here, because the traditional approach that we use is we try to get companies to invest in the expensive part of the drug R&D process, which is the clinical trials. And the problem here is, first of all, we don't know if this is going to work, and if so, for what. So it's not like, a drug where you've got early results that are really promising and you have a pretty clear path forward. It's a real crapshoot. And secondly, the amount of money that you're going to have to pour into this and the diversity of trials that you might even if you focus, that you might potentially have to set up here, is just beyond the scope of what most pharma companies are even contemplating. And at the end of it, what do you get? You get a product that you're going to sell to a small number of people who have these antibiotic-resistant infections, and that's not a lot of a market. This is going to have to require significant government investment and possibly philanthropic investment, but I think government investment, because you already need to get the regulators on board too.
Well, if there's enough pressure, then it will get done. It's right.
Yes.
That's the problem, I think, because right now we still have antibiotics that work.
Mm-hmm.
Yeah.
And
most of the time.
We're not at the crisis point. Right?
Yeah.
For some people, that is true.
Yeah.
So, I didn't go over the stats at the beginning, but the number of people that are experiencing recalcitrant antibiotic-resistant infections is growing. And I think that has really fueled the renewed investment that we're seeing now, not just here in the U.S., but in many other countries. People are very interested in having these alternative options. And shall we say, it's not even... At first, the way that I understood it was that phage were going to replace antibiotics, but I don't think that's really the goal at all anymore. Really, we want phage to help extend the usable life of antibiotics, and that is going to require careful use and not overuse in a way that we end up in the same spot that we started.
And the numbers at the top of this paper, they cite a report that 4.95 million deaths worldwide in 2019 were associated with antimicrobial resistance, and 1.27 million of those were directly linked to antimicrobial. So this is a lot of people dying annually.
So that Belgian study of 100 cases, which you discussed in Australia, TWiV, I remember him saying that the best results came from combining phage with antimicrobials
Mm-hmm.
or antibiotics, I should say. So you can't, as you say, it's not going to replace it.
Did you see my little sample in the show notes?
I
Yes.
saw that. Yes, it's very nice.
Oh, what is that, Rich?
I
It's
don't
a
know.
phage.
It's labeled
Oh, intestiphage.
intestiphage. And
From
the
Eliava
writing on it,
is-
I'm sorry, it's out of focus. But the writing on it, I think, is Georgian.
Yes,
Yeah.
it is.
Okay.
Yeah.
Yes.
This was a
Can't
gift
reach.
to me from Donna Duckworth when she retired. Okay, and it sits on my desktop in a beaker,
Oh, nice. It's because it's an ampule.
along right next to 2 bifurcated needles.
Cool.
In case of emergency break glass.
That's right. This is my little emergency kit.
Tested phage.
They still sell those at Eliava, but not in the ampules. You probably can't see them. Where's my hand? Like, I have a couple on my windowsill up there that I bought when I visited Tbilisi.
So probably you alluded to, the phage is changing. That's the
Yes.
problem with attenuated viral vaccines, right? They
Mhm.
replicate in the host and they change. And, the best example is polio, where the change is a big problem,
Yeah.
right? It mutates, it recombines. So, yeah, this is
And then failed expectations, right?
exactly right. I mean, then it ends up, in the case of polio, it causes the disease that you're trying to prevent, which is really a disaster.
I think overall, I'm encouraged that people are having conversations across countries that can
Yeah.
share their experience, because I think that's the best opportunity for us to make progress forward, is not just trying to work on our own. And that's regulatory agencies, governments, scientists, physicians working together to make something useful happen.
All right. Thank you, Jolene.
Cool. Yeah. Thank you.
Yeah.
So we've been doing some classic papers, and I wanted to do one on the discovery of hepatitis B virus because, it's a classic. As you'll see, we have an interesting paper, but also the Hep B vaccine is under attack these days, and it's not right because it's a fabulous vaccine that takes care of a nasty disease. So we'll put a little attention on hepatitis B virus. So in 1976, the Nobel Prize in Medicine was given to Baruch Blumberg for discovering hepatitis B, for this. And he wasn't even starting out to do this. He was starting out to do something totally different. In fact, in the 1950s, he was interested in whether genetic differences between people could influence disease susceptibility. Right?
And
But in
let's
the 50s,
also in the 1950s, in parallel development, which I guess Bloomberg probably thought of as largely unrelated to his work.
Yeah.
What we knew about hepatitis at the time was that it was a big problem in World War II, and a lot of the troops developed hepatitis. And when we say that, classically, that's a pathology. It's not a virus. So you say hepatitis, it means inflammation of the liver, could be caused by toxins or variety of other things. But you had troops coming down with hepatitis, getting sick, big problem on both sides, all sides. And it became clear during that time, and by the time Bloomberg was working in the 1950s, that there were 2 types of hepatitis that developed in these situations. There was the acute, what they started calling epidemic hepatitis, and then there was another condition that was a little more slow developing and less common, and that was serum hepatitis. And then at some point, somebody decided to call them A and B. And so we had hepatitis A and hepatitis B as diseases, but we didn't know what caused them.
and in fact, he wasn't even interested in that.
No, he
When
wasn't studying that at all. That was like other news.
So and in fact, there's a lovely document, his Nobel lecture. So back in the 70s, they had Nobel lectures, and so you can see the text of it here. So as I said, he wanted to know if genetic differences could account for disease susceptibility, but we had no way of looking at genes back then, of sequences. There was no DNA sequencing. So he said, let's look at serum proteins, because it's easy to get blood from people, and there's some limited things you could do with proteins and antibodies. And he thought he would look at hemophiliacs who got a lot of blood transfusions. See, the idea being they're getting lots of blood from other people, so maybe they're making antibodies against other people's proteins, and they could use those to study these differences. And so, in fact, he had some early evidence that he could find a protein by this method, not related to hepatitis B virus, but it gave him some encouragement to keep going. Now, in 1963, he's in his lab studying New York City hemophiliacs at Mount Sinai. And he's looking to see if you get some antibodies arising in these hemophiliacs that react with proteins from other people. And he finds one patient in New York whose serum reacts with serum from an Aboriginal Australian, right? So far away. And he's like, what's going on here? He ends up calling it the Australia antigen. And they end up collecting serum from all over the world. And he finds this Australia antigen. He calls it rare in the U.S., 1 in 1,000 people. That's not so rare.
That's
right?
not that rare.
That probably was the instance of hepatitis. It's some tropical populations and Asian populations. And then he finds it's common in people with leukemia and people with Down syndrome. So he decides to start looking at, he moves to Philadelphia. He decides to start looking at Down's patients. And he mentions later that these patients are particularly susceptible to leukemia. And that was one of the other conditions that he looked at, right? That people had this antigen.
Yeah, so people think of Down syndrome as causing mental retardation, which it does, and this is a genetic condition. But it causes a bunch of other things too, and including these various types or can cause various types of blood disorders. And so these people are in this study.
So when he's looking at a cohort of Down's patients in Philadelphia, and he checks them regularly. And in 1966, one of his patients, whose name was James, he originally had tested negative. Suddenly, he tests positive for Australia antigen.
But he hadn't been to Australia.
Hadn't been to Australia.
And that's got to be a shocker, because up until this point, I think Bloomberg is thinking that he's looking at genetic differences among people. But now all of a sudden, somebody's genetics changes. No, something's up here.
Something's up, and so they do some studies, and they realize that this James has suddenly developed acute hepatitis. So they eventually follow that up, and they show that Australia antigen, AU, is associated with viral hepatitis. And that is eventually used to develop screening assays for blood products and so forth. So it's really important. So we picked this up with our paper, which was published in 1968. I mean, there are many papers that are part of this story, but this was published in Nature. Particles associated with Australia antigen in the sera of patients with leukemia, Down syndrome, and hepatitis. And there are 3 authors, Manfred Bayer, Baruch Blumberg, and Barbara Werner. And this is from the Institute of Cancer Research in Philadelphia, now Fox Chase Cancer Center. And by the way, Barbara Werner was like his first technician. She went on to medical school. But while she was working on this project, he wrote in his lecture, she suddenly started feeling badly. And she checked her blood, and suddenly she had Australia antigen in it. He says it's the first time Australia was used to diagnose hepatitis she turned out to have. What he called non-icteric, right? So there's no jaundice involved. So often you have jaundice, your eye whites turn yellow and so forth because of liver damage. But she had non-icteric and she recovered.
It's the like nightmare of every PI that the person in your lab ends up with the disease that you study. Even in their case, they didn't necessarily know that it was an infectious disease that they were studying, but fortunately she turned out to recover.
So that's funny. You should mention that, because I just recorded Beyond the Noise with Paul Offa, and he said he was working in Harry Greenberg's lab years ago at Stanford, and Harry wanted to start studying norovirus. So they'd gotten a sample of vomitus from a patient with norovirus. And it came in, and he said the guy opened it right up in the lab without putting it in the hood. And then a day later, off it starts vomiting and having norovirus. And so he calls Harry to tell him, and Harry says, "Did you save your vomit?"
Did you save your vomit?
Because that would have been a nice
Yes.
chain
because
of it.
back then they couldn't grow it, right?
Yeah, still very hard to grow, if at all, right? Anyway, so that's this paper. They go through the history that I've talked about. And then they also talk about some physicochemical properties of Australia antigen. So remember, it's going to turn out that Australia antigen is hepatitis B virus, all right?
Yeah, we already gave you the spoiler.
But I want to tell you some interesting stuff. So they're doing, so there's a technique that used to be used to look at interactions of antibodies with antigen. It was called an Ochterlony plate. And I don't know if any of you have ever done that. Probably Rich has.
I've never done it, but I know what it is.
You take an agar plate, you punch holes in it. So you punch 3 holes, say, you punch in one hole, you put antibody, and in the 2 holes, you put 2 antigens. You don't know if they're identical or not. And then when the antibody and the antigens diffuse through the agar, and where the 2 concentrations are the highest, they make a precipitin band, right? So you can imagine the antibody reacts with an antigen. You get this nice white band, which you can see. And if you have
like with your eyes?
Your eyes, yeah. You can take pictures of it and everything. And so if you have 2 different antigens in the wells, there will not be any cross reactivity. You will have straight lines forming that make kind of a cross.
They intersect. It's like so you get
Right.
this X on the plate where the 2 antigens are ships passing in the night.
But if they have the same antigen or cross react, you won't get a cross, but you'll get a continuous line because they're the same antigen. So that's what they use here. And they say the antigen in these assays, if you stain it it stains faintly with Sudan black, indicating it contains lipid. And that's cool because now we know this is an envelope
Yeah,
virus.
these are the kinds of tests you had to do when you didn't have access to any of this modern stuff.
And it appears in the first peak in a Cephidex G200 column. Oh my gosh. So Cephidex G200 are little beads that you put in a column, and then you can pour things through it and collect them as they come out. And G200 has beads that will exclude anything over, I think, 200,000 molecular weight. It's really big. So if you have a 25,000 molecular weight protein, it will go into the bead, and therefore it moves more slowly down the column. And anything excluded goes right through. So this thing is really big.
I got to tell my Cephidex story.
Yeah, go ahead.
This is from Harry Nuller, because I didn't understand how Cephidex worked. So it's these beads, and they're porous.
Right.
So they have cavities in them. And the cavities are smaller than the spaces between the beads. So big stuff doesn't get hung up in the cavities and goes straight through.
Right.
Littler stuff gets hung up in the cavities depending on its size. Harry said, think of it this way. You have a forest, and you have a mixture of adults and kids, and you tell them to run through the forest, and the adults come out first because the kids climb the trees.
That's very good.
I love that analogy, Rich, because I use it in my class now since you've told it once or maybe twice before.
Okay.
And that's how I tell my students
Absolutely.
what's going on.
I mean, it's stuck with me for 60 years.
So you can buy Cephidex beads with different pores, right? So G25, G50, G100, and the numbers represent the molecular weight of roughly of a globular protein that gets excluded by the pore. All right. Also, they say the antigen's rare in normal people in the U.S., which I already said, 1 in a million. However, it's common in patients with hepatitis, 20%, and they say in whom its presence is transient, so it's not always there. And in a chronic form of hepatitis, as Alan mentioned earlier, they say where the antigen is persistent for many years. It's present in leukemia and in other normal people. So they say, this is very funny. Our view currently is it's an antigen that's in hepatitis, Down syndrome, leukemia, and other people. Yeah, really?
Yeah.
All right. So in this paper, they're doing some electron microscopy and some gradients. They're going to do sucrose gradients to fractionate this serum. Okay. And then, so they'll take serum from patients, they'll put it on top of a gradient of sucrose, run it in the centrifuge, and then they drip the gradient. They'll take drops and they look at each fraction by octolony to see where the Australia antigen is. It's really a kind of tedious assay, right? So they show the figure 1 is actually the gradient, right? They have, and by the way,
I need to interrupt with just one thing here out of the methods. There were several things in the methods that caught my attention, but this is probably the most, in my mind, noteworthy. Their source of antibody for doing these, they say this is in the methods. The rabbit serum was prepared by immunizing a patient suffering from chronic lymphocytic leukemia with the serum and subsequently carrying out absorption of the serum of normal individuals. The actually rabbit serum there doesn't make any sense to me.
No, it doesn't.
Because they say immunizing a patient.
I know.
Suffering from leukemia with serum.
I don't get it
Wait a
either.
minute. No, that can't be right. That's got to be, because I read that, I missed the rabbit thing, and I read that as, man, they're going to give this person hepatitis.
Right.
And get antibody at the same time. But that doesn't make any sense with the rabbit. Something's wrong there.
No, it doesn't make any sense.
All right. Sorry.
But anyway, I wanted to tell you, they have serum from different patients here. They have a patient with leukemia, a patient with reticuloendotheliosis, a patient with Down syndrome, with anicteric hepatitis, and a patient with Down syndrome with acute hepatitis, and another Down syndrome with hepatitis. Okay? So they get serum from them for this analysis. So when they do the gradient, they either load 0.2 mLs or 0.6 ml on the gradient. When you load 0.2 ml, you get a very nice peak in fraction, looks like fraction 11. So remember, they take this gradient and they drip from the bottom, and they take each fraction, maybe every 10 fractions, and do an octolony to see where.
Good times. Yeah.
Yes, right. So it's makes a very nice peak, and if you put more on, it makes a little bit of a broader peak. So they take these fractions from the gradient, so now they're more or less somewhat purified. Right?
Yeah.
And they look at them under the electron microscope, and they see particles. And they say, you could see some structural detail. There's some knob-like subunits of about 30 angstroms in diameter, which would be, how many nanometers would that be? Would that be three nanometers, right? And then they see a central core in some of these particles. And in other particles, the center seems to be filled with stain, so they seem to be empty. Okay, so they're looking at virus particles, and they're looking at empty virus particles and full ones, and the knobs are probably the protein subunits, as you'll see. And then they see elongated particles, same diameter, but like up to 200 angstroms long, and you can see one of them in figure A. And then they could see some particles of 150 angstroms in diameter. And they see these in multiple patients, but when they run serum from normal individuals, they don't see any of these particles. Okay, so now if they add antibodies to Australia antigen, the particles agglutinate. They all stick together, and they have a picture of that as well. So it looks like the particles are made of Australia antigen. And so they say, what does this all mean? If you look at these pictures, you're going to think virus right away, right? They do look, and they must have thought virus as well. But they never say it out. Right? They have, here are the possibilities, right?
Yeah. Yeah, they get their fifth possibility. They finally
they get the virus.
It could be glycogen. It could be particle ribonuclear protein particles. It could be pieces of the endoplasmic reticulum. And they go through and they kind of say, yeah, it doesn't really look like that. Doesn't really look like that. And the fifth thing they mentioned, the relation of these particles to virus particles must also be considered.
Has not escaped our attention.
Has not escaped our notice. Yes.
I think in the lecture, the Nobel lecture, they talked about how people didn't believe them.
Yeah,
like others had suggested that hepatitis was caused by a virus, and they were laughed at. And even when they published this initially, it wasn't believed, and other people had to replicate it before everyone really agreed.
Yep. But they say, these could be picornaviruses. The size is right. Picornas are, about 30 nanometers in diameter, and they go through polio and Coxsackie and echo. They have an icosahedral symmetry. They seem to think these are icosahedral particles, but they actually are, but with an envelope on top of them, not like the picornaviruses. And they say
is HBV one of the viruses that initially it wasn't known that it was enveloped because they always extracted it with chloroform?
I don't know. It's a good question. It
might not be HBV, but I thought it was HBV that the non-enveloped core is infectious. And in the lab, when they grew it, that it might be another virus. Then they could still get it without the envelope, and so it was a long time before they realized that the natural form had an envelope.
Yeah, here they didn't do any extraction, so I don't know. It's a good question.
Well, they also stained it and saw that it had lipid associated.
Yeah. So they also note that for poliovirus, you can see full particles and empty particles, and that's what they're saying. So they say the occasional lack of a core of the Australia antigen particles make it tempting to compare it with viruses of the Picorna group. All right. So then they say, nobody's consistently isolated a virus from patients of hepatitis. So here we have a 25 nanometer particle, which other people have isolated as well. We seem to have particles of different size. And so what we conclude is from these patients, leukemia, Down syndrome, and hepatitis is associated with a particle. And we don't know what it is, but it should be studied. So, in hindsight, you look at this, you go, of course it's a virus, right?
Yeah.
It looks
But that's
exactly
because we know the answer now.
Yeah, that's because we know the answer. So in our textbook, we have a nice picture of the structure of hepatitis B virus. And this is actually based on the picture that he has in his Nobel lecture. And so the native particle, which is an envelope virus, there's a membrane, and within it an icosahedral capsid, and within that the viral DNA. That's about 40 nanometers in diameter. And then there are incomplete particles. There are ones that are about 15 to 25 nanometers in diameter, very small. And then there are elongated particles up to 200 nanometers long, which are the ones exactly that he saw in his electron micrograph in this paper, very long particles. And by the way, these were originally seen by another investigator by the name of Dane. And so for many years they were called Dane particles, right? The Australia antigen forms Dane particles. And it wasn't called hepatitis B virus for a while. But there you have this. You have it's a completely accidental discovery. It was used to make a blood test, which eventually got rid of transfusion-associated hepatitis B, right, which is really good. And then he used it to make a vaccine. He originally purified the surface antigen, which is the viral glycoprotein, from patients and used that to make a vaccine. And it was the first cancer vaccine because we subsequently found that if you give it to babies at birth, it prevents 99% of HBV infections, and it doesn't do that when you give it later, and those babies don't get cancer. So.
But you can also get HBV vaccine as an adult, and it's highly effective.
Of course. And now, of course, we make a recombinant version, so we
Yes.
don't need to purify it from serum of patients.
Yeah. Now, I don't know if you can clarify whether this is acute or chronic infection, but there are some, actually a fairly high titer of particles, but that is virus particles, infectious virus particles, but the S antigen actually swamps that much
Yes.
more of that stuff. And in fact, from an evolutionary point of view, the S antigen is probably like chaff for the immune system, right? It's a distraction for the immune system. So that may be, in quotes, evolutionarily deliberate. But that's one of the things I think that facilitates the purification of this from blood to make a vaccine. It's extraordinarily abundant.
Right. Right. And so if you had, the reason it's a cancer vaccine, as Vincent mentioned, is in cases, and this doesn't happen in every case of hepatitis B, but it can happen that you develop a chronic infection, which then causes the liver to progress to hepatocellular carcinoma. And in areas where this virus was spreading commonly, that was actually a major cause of liver cancer.
So in his Nobel lecture, he has an electron micrograph, a nicer one than in this paper. And then there's a diagram of what he's seeing. And so there's the Dane particle, which is 42 nanometers. That's the complete particle. The Australia antigen is the hepatitis B surface antigen, either forming little particles or elongated particles, as I mentioned, right? You got 20 nanometer spheres or 20 by 200 nanometer spheres. So that's what he was picking up as the Australia antigen. And then in this paper, he sees cores, right? What he calls cores. And that's the DNA, which is with the membrane removed from it. Now, I don't know if that also contains the icosahedral protein shell around it or not. At the time, they didn't know that much about it. But it has lost the membrane. So now it all makes sense.
So all of us who are walking around vaccinated against HBV so we're all immune to the Australia antigen.
That's right, we are. And there's a quote from Blumberg in this lecture. It quote, "This experience does not encourage an approach to basic research which is based exclusively on specific goal-directed programs for the solution of biological problems." I think this is 1970-something, right? He realized that you can solve some things by working on them, but other things you're going to stumble on like he did.
And were, at the time, 1960s, there were lots of people working on the causes of hepatitis.
Right.
Because this was, as I said, it was initially really came to the fore as a military problem. And so the Pentagon was funding a lot of research. What the hell is causing this and how do we prevent it? And nobody had figured it out. And then this guy who's working on the genetics of disease stumbles on this weird thing going on with this Australia antigen. And, here's the cause of one of the two major types of hepatitis.
It's another example of the serendipity of science, right?
Yeah.
And initially, where he was located, they didn't have access to the patients that they needed to be able to solve it. So it even was really lucky that they moved to, is it Philadelphia?
Philadelphia.
Where they had those cohorts of Down syndrome patients that could exhibit that exact infectious disease response that they wouldn't have seen otherwise. So it's a lot of chance and luck, and they were prepared, and they had many years of research before that in the area that they were looking at.
I mean, he was the fact that he also went to Australia, right? Because he was interested in different populations and how they would genetically vary. She went there, and that was an important part of it, right?
Yeah.
To see this cross reactivity between a person in New York and a person in Australia, this is just a great story. So, in light of the attacks on the Hep B vaccine, which are completely unwarranted, by the way, It's very safe, and it prevents kids from getting hepatitis B at a very young age, which can lead to liver cancer. This is how we got there, by this guy stumbling onto it. So it's a very cool story.
So
I love
just
the link that you put in, Vincent, on Hepatitis B Foundation, because
Yep.
the Hepatitis B Foundation was established because somebody got sick with hepatitis, and the people who loved that person were like, wait a second, there's not enough resources for people who have hepatitis. They didn't have internet. They didn't have ways to look up what to do, ways to connect with each other. And it developed into a foundation that helps with not just education, but research and the help of preventing other people from getting hepatitis B and dying from hepatocellular carcinoma.
Yes. Rich can tell you more about the Hepatitis B Foundation.
Yeah, well, that's in my pick last week.
Yep.
Was an interview with Sherry Cohen, who's the current president of the Hep B Foundation, and she goes through all that history. So look back in last week's picks, and you can see that whole thing. It's a remarkable organization. And I just want to reemphasize here for the umpteenth time, and I'm looking at, that prior to universal birth vaccination recommended in 1991, there were about 20,000 children a year that were infected. And and that's actually after a decade. The vaccine was in use since 1981, I believe. And so,
Yep.
but it was applied in a fashion that was thought to be judicious. That is, you didn't vaccinate people, the children at birth who were born of mothers who were decidedly hepatitis B negative, which is what the recommendation now is going to, okay? Going back to the first decade during the vaccine when 20,000 kids a year were getting infected, and 90% of those are going to develop chronic infections and have lousy outcomes. With the implementation of the universal hepatitis B vaccine, that dropped to 20 kids a year, a 99% decrease. And the vaccine is absolutely safe. This infuriates me, I think, more than anything else that's going on. And there's a lot to be infuriated about. But this is going to kill people. It's going to make people suffer. It's going to kill people. This is an indictable offense.
Yeah, I put the link to the foundation is because they have a statement in response to the ACIP decision. And they say, we're very dismayed and concerned about this.
Yeah.
Removing the recommendation for universal Hep B birth dose will undoubtedly lead to more Hep B, more liver cancer cases, and more Hep B-related deaths in the U.S., all of which are preventable. And they go on to talk about the data supporting the current use of it. So you should check that out.
This is not hype. This is not politics. This is science. This is fact. People are going to die.
Yeah.
Yeah, I don't know how these people on ACIP can make this decision without feeling guilty, because the data are all there. There's no reason to change the schedule.
They're not scientists. They think
No,
they're
they're
right.
not.
They don't get it.
Yeah.
They don't understand.
It doesn't matter if you're a scientist and you think you're right. Sometimes you're wrong.
Yeah.
And that doesn't make it okay to make decisions that are going to put people, not just yourself, your family, but many, many other people who don't have control over these kinds of things and don't know. And the worst part is if you know and you recommend not to. And so then that really, I think it's just very inconsiderate, and I don't understand why, but it is a major problem.
Money.
So apparently what swung the committee was this lady, forgot her name. She said this transmission outside of birth doesn't happen in U.S. families. It doesn't apply. Right, so half of the cases in 91 were from the mother, but the other half were from casual contact with other people. She said that doesn't apply. Of course it applies. We have the data showing that it applies, right? So you say that, and now you vote to remove a birth dose. It's dangerous. So this is, no, as Rich said, this is indictable. And if people die, it's going to be a while before they die because this is a long incubation disease. And maybe this whole thing will pass over by then and we'll be back to immunizing. But some people will die from this and we'll see what happens.
Maybe we'll come up with a cure in the meantime. But that's not, that's another whole story. You can beat this thing back so that you can live with it, but it's kind of like HIV. You can't eliminate it, not with the technologies that we currently have.
And if it's any, speaking of the hepatitis B vaccine, they're recombinant vaccines, right? So it's not something that you can get hepatitis from. You cannot get hepatitis from the recombinant Hep B vaccine. There are adjuvanted and non-adjuvanted versions of the Hep B vaccine. So there are options currently, at least here in the US, available to be used that do not put anyone at danger of acquiring a hepatitis infection.
Or any other complication. This is an extremely safe, effective vaccine. Yeah.
Well, fortunately, many medical societies in the U.S. have condemned this recommendation, and one hopes that, pediatricians do the right thing. But, they're going to be parents who say, oh, we don't have to do this, and not do it, and that's the tragedy.
Well, and in the meantime, people are rightfully confused.
Yeah.
And it does damage to the credibility of the health organizations. And that's going to be hard to repair. So
Well, Daniel showed on Clinical Update yesterday a survey of 1,000 people. Only 13% of them now trust the CDC.
Hey, you count me in that one.
I'm in the majority now.
I don't trust them either.
No, I guess it was before the show. I was saying I was looking up some statistics on something because, I'm a science journalist and I have to do this. And among the top hits, CDC information, and I normally that's a go-to. And I thought, oh, crap, I can't. Now I got to dig deeper and find other sources for this information because I can't trust what's coming from that agency anymore.
So I got a text from the CDC the other day from the VSafe program. Do you guys know that?
That's the one that was tracking after the vaccine.
Yeah. So if you got a COVID vaccine, they would track you and they say, how you feeling? You okay, right? And then I got an RSV vaccine last year and that was added to it. So I got a text and it said, have you got a COVID or flu vaccine lately? Let us know. So I click on the link and I have to re-register. And I don't trust them. Why can't I use my old information? It always worked before.
Yeah, what happened to your old information?
Yeah, where is it? Why? I don't trust them. So
Went down the memory hole.
No, they probably screwed it up or something.
Doge probably deleted it.
That was my thought.
They deleted it. I'm not doing it again because I don't trust CDC.
They also probably aren't going to be able to analyze that data because it shows that it's safe. And so what's the
point if they
investing in that?
No one will ever hear about it.
Right.
That's the problem. All right, let's do some picks of the week. Rich, what do you have for us?
Let me see here.
This is
good. Yeah. This is just a little tidbit. So there's a virus discovery dude at the University of Florida, my professional alma mater, named John Lednicki. I'm acquainted with this individual. I've had some even professional interactions with him. I can't quite remember what. But at any rate, he's a virus discovery dude. And one of his methods is his cat. His cat drags in critters of one sort or another. And rather than just bury them or throw them away, Lednicki sees them as sources of potentially new viruses. So he takes them into his lab and does analysis on them to see if they got anything new. And on 2 different occasions now, he's come up with a new virus out of the cat stuff. And this one is I'm not going to. We don't have to go into detail, but this one is the the sample was a shrew of some sort that the cat brought in, and he did the analysis, and it's some sort of orthoreovirus.
Yeah.
Doesn't matter. The best part of the thing is the picture of the cat
Yes.
at the top of the article. Nice looking black cat. So there you go.
He
Never
says at
throw
the end-
anything away, man.
He says at the end, if you come across a dead animal, why not test it instead of burying it?
And,
Well,
and
not
I
everybody
imagine
can do that.
when he's taking this sample and probably putting it in a baggie with gloves on, Pepper is probably looking at him like, "Are you going to eat that?"
Indeed.
Yeah, and he does comment on this that the cat's okay. You know,
Yeah, good. But I have to say, it's a little risky letting your cat go outside in Florida, especially. I mean, you got like pythons eating alligators down there. Right?
Yeah. Well, you got they lose a couple of dogs walking by the ponds every year. Okay?
Yeah.
And so cats are probably quick enough. I don't know. Gators are pretty damn quick.
Outdoor cats are high risk, but we don't let ours out.
I went through this once when we lost a cat. That it was an indoor cat, but I wanted to know what the lifespan of a cat was anyway. And I looked it up and it said average lifespan depends on whether they're an indoor or an outdoor cat.
Yep.
Indoor cats last about 13 years. Outdoor cats last 3.
Yeah.
3?
Yeah,
Did you say
and
3?
thinking back on my own experience,
Wow.
when I was growing up, we had outdoor cats all the time,
Yep.
and very, very few of them lived to old age. They just disappeared.
Or if you do know what happened to them, it's often they got hit by a car.
Yeah.
We had same thing when I was growing up. We had cats in and out at my mom's house, especially, just it's not a safe place to let your cats out. If they are unfixed, so unneutered males, especially, I think it's even less than 3 years because they're just wandering around and picking fights with anything they can, and yeah, big problem.
So our guy now is confined to the indoors. I feel like he's a prisoner sometimes because he would love to get out, but it's a jungle out there.
Yeah.
But my cat loves it outside.
Yeah,
They
She's
do
a free
love it.
spirit. I
Absolutely.
can't keep her in.
Absolutely.
Rich, we got you already.
You
Sorry,
got me.
Alan, what do you have for us?
So I have a video. Actually, this YouTuber sent it to me. He is let's see. He has a PhD in biology of some branch, but he has a YouTube channel called Worm Talk, and he talks about various vermiform things of interest. So that's pretty cool. But I'm picking this particular video because it's very timely. With officials saying that beef prices are going up because of immigrants coming in with their cattle that are carrying screwworm, which is not what's going on with beef prices, and it's not what's going on with immigrants coming in, because you don't bring a cow through a frickin' fence when you're crossing the Rio Grande, and that whole argument is just idiotic. But this is a really good discussion of what screwworm is and why it is spreading north. And this is something I think I've touched on it with a couple of picks, and we've touched on it in TWiV generally. And this is a serious parasite of beef, and it is moving north and has been, spotted up toward the Texas border. But that's not why your beef's expensive.
Wasn't it almost eradicated?
It was, yes. So actually, this is my special interest in this is my grandfather supervised the work that one of his employees, Ed Nippling, developed this sterile insect technique, which is one of the coolest methods for controlling a pest, and has now come back to the fore with genetically engineered mosquitoes and such. But what you do is you release a bunch of sterile male flies. They mate with the females. The females then lay eggs that will never hatch.
Mm-hmm.
And you keep the way they classically sterilize these flies was just irradiate them. So you breed up a whole bunch of flies, irradiate them, release only the males. You get, generation after generation of the sterile eggs, and that reduces the population. So now your released flies are a bigger proportion of it, and it kind of spirals on downward, and you can actually drive them to extinction locally that way, which is exactly what we did all the way down to the Darien Gap in Central America, which is like the narrow part of the isthmus right near the Panama Canal. We couldn't get it any further south than that, because then you get into South America and it's just too hard to control on that scale. And for decades, screwworm, Hominovorax cochleomaya, it's a fly. It's not a worm, by the way. So what happens is the fly lays an egg in any kind of a wound on the cow, and then the egg hatches into a maggot, which eats into the skin, and this makes the cow sick. And so you get decreased beef yields. But we kept it contained through continuous, USDA funding to Central American countries and Mexico to continue these sterile fly releases. And now, because of a variety of factors that are covered in this video, that control is weakened, and now, beef cattle are getting smuggled and things are getting out of control again.
I'm just looking at this. Sorry.
We're building, they call it a factory, to produce these screwworm males that can be sterilized to release here in Texas because of how big of a deal it is that it's coming back close to Texas. With all of the cattle industry that we have here, it's a major concern. So they're trying to very quickly in a short period of time build the capacity to make more, because we don't have enough to keep pace with the rate that it seems to be spreading.
Wow.
Yeah, back when this was an endemic disease in the U.S., it was a huge, constant loss to cattle farmers.
Who is this guy? The video looks really good.
I got his
says
name
he's got
in
a
my email
PhD
somewhere.
in biology.
Yeah,
and he likes talking about.
Yeah, and I recommend his
and,
channel generally. It's
but
anyway,
I can't
Worm Talk,
find
I'm
out
sure you're listening. Send another note
Yeah.
with your name to the show.
Yeah,
We'll cite you
that.
by name.
Thank you. Jolene, what do you have for us?
Yeah, so I'm picking a book. This is Aerosmith by Sinclair Lewis, which I have here in my office and that I still occasionally give to undergrads. It's considered to be a fictional account of, I consider it to be a fictional account of Felix Darrell and the discovery of phage, and it kind of goes on to this idea of the holy grail of doing a randomized controlled trial in plague, and it's really kind of a social commentary on the culture of science at the time that it is set in. It's a really, I found it to be a really nice read, and kind of gives you the personal side of eccentric scientists who are really just interested in all sorts of things, and it kind of goes through the character's journey of studying to be in medicine and then research and then moving up into New York. And I think it's meant to be a kind of a fictional similarity to the Rockefeller Institute.
Yep.
And it then also goes into, there's some human side in terms of there's this very loyal wife, and then some things happen to the relationship at the end. But I think it talks about kind of self-funded science as well. And it's a really great story, a fun read. So Aerosmith by Sinclair Lewis.
This is an absolute classic. This is,
Here's
I no longer have my copy, but it's. If
So
you're
mine
in
is-
anything related, read
My
it.
copy is from 1961, and my mother used to teach this in English class, so it's full of all her writings. You can't really see it.
Oh, wow, cool.
Very
But all her
cool.
notes on the sides of the pages. And she's the one who turned me on to this many years ago. The McGurk Institute, supposedly Rockefeller. Yeah, it's great. It's a good story.
And it's a quick read too. It's
Yeah, you have to know what happens.
yeah.
All right, my pick is a column by Jeremy Faust. Jeremy Faust is an ER doc who also does science communication, and he has a story that just came out yesterday. It's in Inside Medicine. It's actually his Substack called Inside Medicine. And the column is FDA vaccine chief's memo cited 10 pediatric COVID-10 vaccine deaths, but the agency's own analysis found between 0 and 7. So remember, last week or the day after Thanksgiving, Vinay Prasad said, I have evidence that the COVID vaccines killed 10 children in the U.S. And apparently this is not true because there's now been an internal review by FDA scientists which leaked, and they say that 0 deaths are certain to have been caused by COVID vaccines. 2 were probable or likely, and 5 were possible. So he...
Do we know how many total COVID-19 vaccines are administered in the U.S.?
138 million. 138 million doses to children 12 and under only. It's in this column here. Very good. But anyway, it's a great article on why Prasad, he basically jumped the gun. He didn't wait for the analysis to be done. And he released what was supposed to be an internal memo, but of course it leaked. And it's now contradicted by this internal review, which I presume we'll hear about more in coming days.
Or which will be completely suppressed.
Right. Interesting, Dr. Prasad did not wait until the FDA scientists he assigned to work on the topic had completed their task before firing off a memo to the staff of his center. Anyway, it gets more crazy every day. Anyway, he's got some other good columns here, too. You should check them out. Inside Medicine by Jeremy Faust.
And he calculates, so based on the data, that even if you took all 7 of the possible likely deaths as having been caused by the vaccine, it's lower than the rate of the per dose risk of anaphylaxis death from ibuprofen.
Right.
Yeah.
All right, we have a couple listener picks. One is from David. I submit for your consideration another astronomy pick of the day from 12/8/2025. This is actually a YouTube-hosted video set to music capturing views of the Earth from the ISS in 2011. Beautiful and haunting in a way, especially when you see the thin atmosphere on the horizon. I think this is a planet worth preserving for future generations, but as an inhabitant, I'm somewhat biased. Thank you for all you do to communicate what science is about. Wishing you all safe and happy holidays and a movement back towards sanity in 2026. Let's see.
This is a beautiful video.
Gorgeous.
Yeah.
These are the kinds of things that make me think like, oh, wouldn't it be interesting to apply to be like an astronaut?
You could. That's what's her name, Rich?
Kate Rubins.
Kate Rubins.
Yeah.
Yeah, that's what she did. And also, Eric writes, if you can't get Taylor Swift to write a pro-science song, you might see if you can get Jesse Wells to do so, since his songs cover a variety of timely political topics. His Wikipedia page points out that he's been described as a modern Woody Guthrie and gives a link. But he also reminds me of Bob Dylan. Here's his song, United Health, from December 2024. All the best, Eric. Thank you, Eric.
Yeah, I recognize this guy. His videos have come across my desk before, I think. And, actually, he looks like Val Kilmer. A little bit. Oh, darn, I'll have to listen to this. Thanks. That's good.
And that'll do it for TWiV 1279. You can find the show notes at microbe.tv/TWiV. You can send your questions or comments to twiv@microbe.tv. And if you enjoy these programs, we would love your support. Microbe.tv/contribute. Jolene Ramsey is at Texas A&M University, ramseylab.vercel.app. Thanks, Jolene.
Thanks all. Glad to be able to discuss these important things together.
Alan Dove is at alandove.com, turbidblack.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. I look forward to being with you guys. This is good fun.
I'm Vincent Racaniello. You can find me at MicrobeTV. I'd like to thank the American Society for Virology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.